Okay, I'd like to welcome everybody to the kickoff of this year's Timothy Johnson Medical Scholars Series at the Virginia Tech Fralin School of Medicine. It's great to see the students here and the ones that are on Zoom. And in a moment, Dr. LeConte will have the honor of introducing today's first speaker. But I just want to take a moment, since we're starting off the season, and say a little something about the program. And so the history of this program, the Timothy Johnson Medical Scholars Series, is such that Dr. Timothy Johnson, who is now sadly deceased, was one of the founding faculty members of this medical school. And Dr. Johnson was a very firm believer in the importance of biomedical research and was really integral in the earliest days of planning and the implementation of the program here at Virginia Tech Fralin School of Medicine that has research as one of the, as you all know, of course, one of the value domains and an integral part. So we had the pleasure before Dr. Johnson passed that he was able to actually come back when he was quite ill at the time, actually, and see the first seminar honoring him in his name. And I can tell you, we were all very pleased to see how he was touched and his family was touched by that. And so it's a really appropriate recognition. And it is really a great honor and pleasure to have the kinds of folks come in that you'll certainly hear today and going forward, who are themselves physicians who practice medicine, but are also outstanding scholars and contribute to discovery in the medical research process. We like to think that some of these folks are good role models for many of you and will provide opportunities for networking in the future and so forth. So I'm sure you're going to be delighted with today's presentation. So without further ado, I'll turn it over to Dr. LeConte, who will introduce today's speaker.
Thank you, Dr. Friedlander. So today I'm very excited to have with us Dr. David Fajgenbaum. I first heard him speak at a AAMC meeting in 2018. And his story was incredibly inspiring and exciting. And so I knew I wanted to bring him here, and it actually wasn't because of all of those sort of inspirational stories that you can see on YouTube. He's had stories written in the New York Times and NPR. So that's certainly a really important piece of why I wanted him to come here. But really, it was about his approach to research, because I thought it was so applicable to what we're trying to do here at VTC with both our medical students and our graduate students. So this series typically has, it's a medical scholar series, and we typically invite people who have sort of long storied careers in research. So they've published, hundreds of publications. They're sort of known throughout the world for their particular area of research, and they've been doing this for decades. So Dr. Fajgenbaum doesn't necessarily fit that description. He graduated from UPenn's Perelman School of Medicine in 2013. So that's a relatively short time ago. In that time, though, he's already published or been collaborated on 21 peer-reviewed scientific papers, 14 peer-reviewed review articles. So he's already had an incredible impact in a really short period of time. And I think there's two particular reasons for that I wanted our students to hear about. So the first is the fact that he really took advantage of this approach that involves collaborating with patients who are experiencing the disorder that he's interested in. Now, obviously, there's a personal bent in this particular story, but I think for our students going forward, remembering that the patients who are actually experiencing this disorder are often at least as expert as any physician or scientist is. And so sort of marshaling that expertise is a particular theme that I want everybody to sort of pay attention to throughout this seminar. The second aspect, I think, of Dr. Fajgenbaum's research that really makes him appropriate for this particular venue is the fact that his research was built on years and years of meticulous research from the basic science world. Not that he was particularly involved in, but he was able to turn to years of descriptions and characterizations of signaling pathways that allowed him to use that basic science and quickly translate it into something that had a huge impact on a patient population. So those are both of those sort of arms of research approaches, the collaborative research with patients and the building upon basic science research, I think is something that our VTC and TBMH students in particular are going to be well poised to do in the future. So I want you to listen to this seminar and think about that, and then ask some questions at the end. And with that, I'm going to go ahead and turn it over to Dr. Fajgenbaum.
Well, thank you so much for that introduction, Leslie. It really was special for me to spend the last hour with a number of students who are there today. I got a chance to do a lunchtime chat with about 13 of you guys, and so it was a fun start. And I hope that that my talk today is able to hit on some of the goals that Dr. LeConte mentioned. And I really hope that it'll be helpful for all of you as you continue in your journeys towards either becoming physicians or scientists or physician scientists. So I'm going to start by sharing my screen. Great. So I titled my talk Chasing My Cure, and I think that'll become quite clear to you all quite shortly around why I say chasing my cure. It's been a journey that I've been on to search for treatments and maybe even one day a cure for a disease that I'm personally facing. But just as was mentioned earlier, it's really not just about chasing my cure, but it's about what we can learn from chasing my cure that can help with chasing many, many more cures. And so I've written a book that's titled Chasing My Cure, but I do often say that I really think that we should have called it Chasing Our Cures, because though I initially set out on this journey to find a treatment that could save my life, really we've been able to make an impact far beyond my life. And one of the reasons we were able to make an impact beyond me and make the impact that we've been able to make is really because we brought together 5 different perspectives. And I think this really hits what Dr. LeConte was referring to. And that's that it wasn't just me as a physician or me as a scientist working to try to make progress. It was a recognition that we needed caregivers of patients. And this is partly informed by the fact that I myself lost my mom to cancer while I was in college. And so having the experience of a caregiver made me realize the importance of a caregiver when we think about taking down and battling diseases. As a medical student, you'll soon hear that I was a medical student, kind of in a very close situation to where you guys are right now when I personally became ill with Castleman disease. Recognizing the voice that needs to come from being a patient and how patients are often left out of the narrative. Recognizing that if I wanted to survive and I wanted to continue to take on diseases, that I would need to become a scientist, and that it couldn't be just kind of my physician hat or my patient hat or even my loved one hat, but being a scientist would be critical. And really lastly, realizing that even all of those 4 perspectives are really important, but you also really need advocates and connectors who can help to bring those together, because physicians, researchers, patients, loved ones, none of those people have the role or the job description of connecting people. And so I realized that there's this 5th role that's really critical. And I'm sitting here today chatting with all of you because of each of these 5 roles. And what I hope you'll take away from this is that as you move forward in your careers, make sure that you include people from all 5 of these realms in the work that you do. So just to highlight again, it's the physician, it's the patient, it's the physician, it's the advocate, connector, the researcher, and the loved one. And when you can connect all of those individuals on an individual level, you can really make a tremendous amount of progress. What I've also learned is that it's not just about chasing my cure. It's not just my individual level, but you can learn so much when you look within a given disease. So when you start connecting patients within that condition and across these various stakeholder groups within a given disease, and then even more importantly, across diseases. So lessons that we learn for Castleman disease that we can share with the ALS community and vice versa. This is where we really make the progress that we need. So I thought I would start back with way back to right around the time that I graduated from high school. This is a picture of my mom and I on my high school graduation day. And at this stage, I was going to Georgetown. I had been given the opportunity to play football at Georgetown, which was really kind of my lifetime dream, was to play college football. And shortly after the picture was taken, I headed off to college, And unfortunately, a few weeks after I got to Georgetown, I received a phone call from my dad where he notified me that my mom had been diagnosed with brain cancer, a very, very aggressive form of brain cancer called a glioblastoma. And so I rushed home immediately because I was just so devastated, and I wanted to be there for the surgery to resect the tumor. And I remember getting to Duke, where the surgery was being performed, and passing by this wall. And it says, "At Duke, there is hope." And I remember getting a tremendous amount of hope from just knowing that I was in the place. I was at Duke. This is the oldest brain tumor center in America. It is really a leading center for brain tumor research. And I really felt like we were in the right place. And I could just hope and pray that my mom would live longer than all of the odds were telling us. This is a picture of my mom and I not too long after her tumor was taken out. And she taught me so much. She battled brain cancer for the next 15 months. And during that time, she taught me about how to battle with grace, how to fight as hard as you can, but do it gracefully, how to really, fight a battle where there maybe is no hope, but with hope. And so I learned so much from her, and her experience also led me to say, okay, I knew I was always interested in medicine, but I'm not just interested in medicine. Now I'm interested in oncology. I want to treat patients like my mom. Unfortunately, she passed away about 15 months after her diagnosis. And I just knew that I wanted to go into medicine. I wanted to take revenge on cancer and on diseases like cancer. And fast forward a little bit, I went to graduate school at Oxford, where I studied cancer prevention. And then I came back to the States for medical school at Penn. And this is a picture of me during my third year of medical school at Penn, when I was making progress towards my goal of becoming an oncologist one day, and just so excited to be in the clinic seeing patients. And out of nowhere, over the course of just a few weeks, I went from being this healthy third-year medical student to having my organs shut down. My liver, my kidneys, my bone marrow, my heart, and my lungs all started to shut down. I gained 70 pounds of fluid. I drifted in and out of consciousness. I required blood transfusions daily to keep me alive, all with no diagnosis. And here I was at UPenn's hospital. I'm biased, but arguably one of the best hospitals in the world. And no one could come up with a diagnosis. And I was just suffering kind of unrelenting pain. My organs were shutting down, and I was certainly on a trajectory where I would likely soon not make it. And about 7 weeks into this, I really hit a low point where I just did not have any more battle in me. I felt like I was, there was no diagnosis. I was getting worse and worse and worse. My family was suffering. I was suffering. It was just a really, really low point for me. And I want to share this low point with you all, because I think it's important to know that we all go through low points. And I'm here today, and obviously have been fortunate in a lot of ways. But I did want to share my absolute lowest point in my journey was when there was no diagnosis. I was, nearing death. And there was a moment while I laid in that hospital bed, and I saw the telephone in the room, and I saw the telephone cord, and for a split second, for maybe even less than a second, I thought to myself about, maybe I should just kind of speed up the inevitable. Maybe I'm not going to survive. My family's suffering. I'm suffering. Maybe this telephone cord needs to speed up what's already going to happen. And as I mentioned, it was within a split second, thankfully, I kind of came to my senses and realized that, no, I don't need to do anything crazy and, try to end this suffering before I need to. And, maybe I can keep fighting. And really the main reason that I changed my mind and thought that way is that I couldn't help but think about my family and what they were going through, what they would go through. And so I decided to keep fighting. And thankfully, right around the time that I decided I was going to keep fighting and keep, trying to make it through every day, a well, at this stage, a lymph node biopsy was finally done. And we'd hoped that maybe this lymph node biopsy, so taking out one of my lymph nodes, looking at it under the microscope, would maybe give us a sense for what was going on. The doctors thought that I likely had lymphoma. And maybe if we had a diagnosis of lymphoma, we could figure out what drugs should be used. But I kept getting more and more and more sick. In fact, I was so sick that a priest came into my room and administered my last rites to me about 11 weeks into this. And I've really considered that moment of when my last rites are read to me to really be the start of my overtime. And many of you all will know what I mean when I say overtime. It's time that you didn't think you'd have. It's extra time, but it's time where every second really has to count. And so really informed by the feelings that I had before going into overtime and recognizing the importance of making every second count, I wanted to read. And throughout my talk today, I've got a few passages from my book that I want to highlight. So the experience of being in overtime is surprisingly one of intense awareness and scrutiny and clarity. There's a reason for that. When there are only a few seconds left on the board, all distractions disappear and the purpose victory becomes clear. The present is only the things around you, and overtime is all present, nothing but present and purpose. And so really ever since that moment when I had my last rites read to me and I decided I was going to keep fighting, I've really felt that I've been in overtime and really tried to make the most of every second. And I mentioned earlier that I played college football. And so I know a little bit about the experience of playing in football in overtime. Although unfortunately we lost a lot more games at Georgetown than we won, or that even went into overtime. But I certainly did and can appreciate what it means to be in overtime. It can be really scary to be in overtime. You know, every mistake is accentuated and could result in the game being over, but it can also be clarifying. Okay, I'm in overtime. I'm going to make the most of every second. I'm going to be intentional about everything that I do. And I've tried to do that ever since. In college, I also met my girlfriend, Caitlin, and we started dating before I went off to grad school in England. And fast forward a few years to when I was a medical student at Penn, we had dated for several years and broken up just a few months before I became sick. And when we broke up, we both felt like we have got all the time in the world. We're 25 years old. If it's meant to be, it's meant to be. And then there I was in ICU having my last rites read to me, realizing that actually I didn't have all the time in the world. And so I made a commitment to myself as I laid there that if I was going to survive, and if I did survive, I would live with an overtime mindset. And in overtime, I would think it, do it, meaning that every thought, like I wish we were together, I would actually do what I could to make that happen, as opposed to just saying, oh, if it's going to work out, it'll work out. So I think this is an important passage to highlight. It came from the very simple realization that the regrets that ate at me the most as I prepared to die weren't about anything I'd done. My greatest regrets were things that I had thought of doing, but never did. It's the absence of memories that you wish you had made and the realization that you'll never get the chance to make them. So thankfully, right around this time, the lymph node biopsy results came back. So this is a picture of my lymph node. It's an H&E stain of my lymph node where the pathologist looked at it and they said, this looks like idiopathic multicentric Castleman disease. So for idiopathic MCD, most patients we think have lymphoma. They get very sick. They're often hospitalized in the intensive care unit, but the lymph node biopsy comes back not as lymphoma, but as being idiopathic multicentric Castleman disease. Well, what is that? It's a disease that really sits at the intersection of cancer, autoimmunity, and infectious disease. We don't know why the immune system gets out of control. Maybe it's a lymphoma. We just haven't figured it out yet. Maybe there's some sort of somatic mutation that leads to this hyperinflammatory response, or maybe it's an autoimmune condition, or maybe there's some sort of pathogen that is stimulating the immune response. But up until now, we still don't know why idiopathic MCD occurs. But we do know that this Castleman disease lymph node is really critical. So you cut the lymph node out, it has a certain appearance to it, which can sometimes be localized, what we call unicentric Castleman disease, or it can be in multiple regions. So you can have multiple regions of enlarged lymph nodes. That's what we call multicentric Castleman disease. It can be caused by a virus called HHV8. Can actually cause your immune system to get out of control, and it can cause multicentric Castleman disease. And there's a form of multicentric Castleman disease that's caused by a cancer, what's called Poem syndrome. So a clonal cancer cell population ignites the cytokine storm, and wreaks havoc all throughout the body. But idiopathic multicentric Castleman disease means you get this cytokine storm. So liver failure, kidney failure, flu-like symptoms, bone marrow shutdown. You get this incredible immune hyperactivation, but we don't know why. There is no virus that we can find, and there is no cancer that we can find. So we call it idiopathic MCD. About a third of patients with my diagnosis die within 5 years of diagnosis. Another third will die within 10 years of diagnosis, making it about as deadly as when you look at all cancers combined. About a third of patients will die within 5 years of diagnosis among individuals with cancers. And as I mentioned, it kind of has overlap with autoimmune conditions, cancers, and infectious diseases, and we don't know what causes it. But thankfully, with the diagnosis came treatment, and I was immediately started on chemotherapy. Thankfully, the chemotherapy was given just in time, and I began to improve. You can see my dad and I here. My head's bald from chemotherapy. My dad's supporting me. And in this picture, you can appreciate how large my belly is. So because of the liver and kidney failure, I gained a tremendous amount of fluid in my belly. But this picture on the right is from New Year's Eve 2010. And in this photo, I'm just so thankful to be alive, frankly. I've got a huge belly because my organs aren't working, but I'm just happy to be alive. So the picture on the right from New Year's Eve, my dad and I at about 8 p.m. went for a walk around the hematology oncology floor at the hospital, and we passed a gentleman who looked like he'd been drinking on New Year's Eve. He was kind of like swaying in his chair. And on our next lap around the floor, we saw that he had fallen on the ground. And so my dad helped him back into his chair. He looked at my dad and I, and he said, "Thanks so much. Good luck to you and your wife." We're like, "Wife? What is he talking about?" And then I looked at my belly and I realized that he thought that I was my dad's pregnant wife, which was a low point emotionally for both of us. I turned to my dad. I said, "Man, Dad, you've got an ugly wife." And we laughed so hard. And I think that I want to highlight an important lesson here. And that's that during times like this, you may think that laughter is the last thing that you'd want to do. And you may think that for your patients, laughing about something like this is kind of crazy, but actually facing my horrible moments with laughter was just as fundamentally a rejection of Castleman's dominion over me as anything else I was doing. It cleared my mind. It stiffened my resolve. It was entirely up to me to determine what was and what wasn't funny. Perhaps most important, humor is social. For me and my family, there was never a better way to reset our collective resolve than laughing together. And you can kind of see how someone maybe would have thought I was a pregnant woman. I have a huge belly from all the liver and kidney failure, and I didn't look like myself. This is a picture from a few years before when I played college football at Georgetown. And I always say this is like the worst before and after picture or pictures of all time. If we could flip the order, it could be this like great advertisement for Peloton, for Muscle Milk. But unfortunately, it's in the wrong order. So I took a picture every week for the next 8 weeks, and you can see my hair starting to grow back. You can see my belly starting to go away. And then pictures from the side, of course, as well. And again, hair growing back and belly starting to go away. So when I got out of the hospital, I immediately reached out to Caitlin, and I was so thankful to learn that she also had been wishing and hoping that we were still together throughout my journey. And we started dating again. This is a picture of the two of us at the next Halloween. I'm dressed as Borat in this picture, but we started dating soon thereafter. And we really committed that we weren't, at least I committed that I would no longer just say, okay, if it's meant to be, it's meant to be. We'll deal with that in the future, always trying to make the most of every moment. This is a picture that I think is really important to me, and that's because on one of my shoulders, I have a bag of chemotherapy getting infused into me, and that's chemotherapy that we were hoping would keep me in remission. And on my other shoulder is Caitlin supporting me and helping to keep me upright. And I think it's really fitting for how things were going at the time. So I was able to return to Penn for medical school, and was hopeful that my disease would be in my rearview mirror, that Castleman disease would not come back. I was on an experimental drug that was undergoing a randomized controlled trial, and I was hopeful that this drug would work and that I would be in a long-term remission, and then I could go back to my plans of becoming a clinical oncologist. I did get involved in the Penn Orphan Disease Center. So orphan diseases are just another term for rare diseases. Got involved in ODC. When unfortunately about a year later, I relapsed. And so this is now for the fourth time I became incredibly ill, and for the fourth time I almost died. The difference is that this is the first time that it happened while I was on this experimental drug. So now I had failed to respond to the only drug in development for my disease, and there were no more options. And so I promised my dad, sisters, and Caitlin that I would dedicate the rest of my life to trying to find a solution, a drug, a treatment, something that could help me to survive. And so I began conducting laboratory research at Penn, and I also started a foundation called the Castleman Disease Collaborative Network to really accelerate research on a global scale. And as Dr. LeConte mentioned earlier, I'll share a bit about the process we take and why it is unique and why it is being replicated by other groups. But there's a really important moment right before I decided I was going to take this disease on. And that's when I realized that I'd been hoping and praying that some researcher somewhere would figure out a drug that could help me. And then there was a moment where I realized that if I was going to hope and pray for someone somewhere to find a drug for me, I should turn that hope into action. I should actually do what I can to actually make that hope become a reality. And so this is a passage I wanted to highlight. Hope cannot be a passive concept. It's a choice and a force. Hoping for something takes more than casting out a wish to the universe and waiting for it to occur. Hope should inspire action. And when it does inspire action in medicine and science, that hope can become a reality beyond your wildest dreams. So thankfully, I got chemotherapy with my 4th episode, and thankfully it saved my life for the 4th time. And when I got out of the hospital, I wanted to understand, well, what's going on right now for idiopathic multicentric Castleman disease? So I learned that about $10,000 a year was being invested into translational research to look for new drugs and new approaches, which is just so little money in the grand scheme of things. The NIH had never given any money to study IMCD. There were no patients in a registry because no registry existed. There were 2 advocacy organizations that existed, but there was no sort of unifying plan or research infrastructure in place. There were no cell lines, animal models, biobanks, registries. The disease was very poorly understood, as you can imagine, as a result. There was no diagnostic criteria to diagnosing the disease. You had to hope that the right doctor had read case reports or remembered it from medical school, but there was no criteria or checklist. There were no treatment guidelines on how to best treat the disease, what chemotherapies could be used. There were no blood tests you could run to predict whether you're likely to get better on one drug or another drug. There were no FDA-approved treatments, and the one drug I mentioned that's in development had not worked for me. So back in 2012, it was terrifying being in the hospital. It was almost even scarier being out of the hospital and understanding the state of research at that stage. So just to kind of highlight where we were scientifically, many years before, we had characterized and understood that Castleman disease involved flu-like symptoms, liver, kidney dysfunction, bone marrow dysfunction, and these abnormal features in the lymph node. That was well established. What wasn't so well known is, well, why is this happening? Why is the cytokine storm happening? Well, back in the 90s, a researcher in Japan named Kazuo Yoshizaki discovered that interleukin-6, which is one particular cytokine. Cytokines, I mentioned earlier, are produced by immune cells to help to communicate between immune cells, but also for effector function, so to help to fight off viruses, bacteria. So we learned that interleukin-6 was very elevated in a portion of patients. But even though we knew interleukin-6 was really important, we still didn't know why it was elevated. We didn't know what intracellular signaling pathways were active or responsible for the elevated IL-6. We didn't know what cell types were really critical and involved. We didn't know the etiology for why the interleukin-6 was elevated in the first place. But even still, we knew that interleukin-6 was important. So drugs were developed like cetuximab that block interleukin-6 directly. And amazingly, we learned that about 1/3 of people just get totally better if you block interleukin-6. So that drug that I was on that I mentioned earlier, I got this drug, cetuximab. It didn't work for me. We learned that it works for about 1/3 of people. So for people like me, where interleukin-6 is not the problem, now we don't know the cause, but we also don't even know what cytokines are the really important cytokines. So for patients like me, we use cytotoxic chemotherapies that basically just destroy as many immune cells as possible. And in doing so, that can actually save patients' lives like my own. Unfortunately, it's not a very sustainable approach. And many of us, in fact, almost all of us, will relapse despite the chemotherapies. I did want to highlight the person who discovered interleukin-6 as being important in Castleman's. This is Kazuo Yoshizaki, who did the work in the 90s. He's a good friend and collaborator of mine. And Kazu both discovered interleukin-6 to be elevated. He also did research to develop the drug that targets the receptor for interleukin-6. And then I had heard a rumor that he had tested it on himself. He had actually given it to himself to prove that it was safe. And so I went to Kazu and said, Kazu, Makoto just told me that you gave tocilizumab to yourself to prove that it was safe. And he said, no, I didn't give it to myself. The nurse, she gave it to me. And I said, exactly, Kazu. So Kazuo Yoshizaki he made the discovery, developed the drug, tested it on himself, and proved that it was safe, went on to test it in other patients. Importantly, this drug tocilizumab is used in individuals who get what's called CART therapy. It's a type of cellular immunotherapy for cancer that creates a Castleman's-like cytokine storm. So patients that get these cells reinfused in them get really, really sick, like they have Castleman disease. And doctors at UPenn, where this was pioneered, gave tocilizumab, gave this same drug that Kazu developed, and it saved the first young patient's life. This is Emily Whitehead, the first patient to experience this. And it saved her life. And thankfully it did, because it would have completely halted the whole cellular therapy program if this first pediatric patient had died without getting the drug that Kazu developed for Castleman disease. So Castleman's is a rare disease. It's only diagnosed in about 5,000 patients each year in the United States, which makes it about as common as ALS. But this is an example where, though Castleman's is rare, a Castleman's drug was now critical for keeping an entire cancer research program alive. So, and then more recently, we've seen the same drug, tocilizumab, used all across the country and the world for COVID-19. Unfortunately, the data is actually quite confusing, and it's actually not that clear now that randomized controlled trials have come out as to whether this drug actually even works. There's some sense that maybe it works in a subgroup of patients. But again, example of a drug developed for a rare disease like Castleman's being used in more common diseases. So I wanted to highlight the research approach that we've taken. In order to highlight our approach, I thought it was important to highlight the way that things are typically done. So in research, patients usually raise money, and then research organizations will invite people from the research field, so physicians, researchers, to apply to use that money however the physicians and researchers think the money should be used. And then a small fraction of the community will actually apply for funding, and an even smaller fraction will be awarded that funding. And so you're getting ideas from a fraction, and you're funding a fraction of a fraction. Study happens, and then 6 months later, a year later, whatever it may be, you fund another round of studies. And so this is a very kind of, it's a parallel process where things are happening in parallel. We decided to create a bit more of a circuit. So in this approach, you're hoping the right researcher applies for the right project at the right time. We wanted to create a bit more of a circuit that would be more likely to lead to breakthroughs. So the first thing we did was to build a community. And so we identified all the physicians and researchers worldwide. We connected them with patients and also physicians and researchers from related diseases in an online platform. And then we started crowdsourcing the whole community. So not just saying, we have money, who wants to apply for it? We said, we have money, we need to ask everyone what research ideas you have, whether you can apply to do the work or not. Just give us all of your ideas for what research needs to be done. And amazingly, we got a lot of different research ideas. Then we were able to prioritize what are the most important studies, and then we were able to reach out to the best researchers to do those studies and to recruit them into the field. So you get more ideas and you get more people that you can go to get the work done. Then we would fundraise based on the studies we had to do. Then we would get samples and data, and we perform the studies, we perform analyses, and we would share that data back with our community. One thing I would highlight about this in addition is that as we learn more about the disease from our scientific experiments and our studies, we always ask the question, what drugs are already FDA approved that may have an effect on this based on what's already known historically? So a drug that may be approved for one thing, but could that drug actually have an effect in Castleman's because of the way that it works? I knew that this wasn't something I could do on my own. And so I knew that I wanted to connect with other patients. So I held a webinar and we invited a bunch of Castleman's patients to join on board. We showed this picture of a Castleman. Castleman disease is named after Benjamin Castleman, but we've kind of repurposed the name Castleman to be a Castleman, like this image. And it's also like this, what I have on my lapel. And so we held this webinar, and about 2 weeks later, this showed up on our Facebook page. One of our patients got it tattooed. He screenshotted the photo, got it tattooed on his shoulder. Now there are literally hundreds, maybe even thousands of Castleman tattoos, but it shows that these patients are just waiting to get behind something. They wanted to be engaged. And then we do something called the Castleman Flex, which if we were all in person today, at the end of the talk, I would have all of you guys stand up and do this Castleman Flex, but maybe you guys will do it on your own and use our hashtag for Castleman Flex to help raise awareness. So I got a huge number of classmates from medical school and also from business school to help us out, because again, it's not something that I could have done on my own. We've been able to build a large community of physicians and researchers from all over the world that have both contributed data, samples, supported patients, treated patients. They're all part of our network. And I wanted to highlight that if we really wanted to turn the tide against Castleman disease, we needed to expand our effort to include more than just those directly affected by it. More people needed to learn about it so it wouldn't continue to be one of the most deadly, most common diseases that most people have never heard of. We wanted to change that so that more people were aware of it. And in the midst of a lot of progress and really driving forward the program and the science, I graduated from medical school. This is a picture of Caitlin and I on my graduation day back in 2013. And shortly thereafter, I started business school. And unfortunately, I relapsed in the beginning of my first semester of business school. And I had just gotten engaged to Caitlin shortly before that. And this is now for the 5th time that I was so sick and nearly died battling Castleman disease. You can see I'm bald here because I got chemotherapy again. And the chemo really kind of just barely saved my life. So I want to highlight what we did at this stage. And so the first thing we did was look back to see what treatments had we tried, what can we learn from those treatments. This graph is a little complicated. I think you guys might have seen a similar version of this in the paper that we sent around. But basically the vertical dotted lines represent when I was first hospitalized. You can see there's C-reactive protein, which is a marker of inflammation, albumin levels, platelet counts, and you can see there's a lot of spikes and valleys in the first 6 months after diagnosis. I was given steroids, I was given rituximab, I was given the IL-6 blocker, siltuximab, but it wasn't until I got chemotherapy that I got a year-plus long remission. Unfortunately, I relapsed again on siltuximab, and again, I needed chemotherapy to give me another year-long remission. But unfortunately, I relapsed, and now for the 5th time, I was had nearly died from this disease, and chemotherapy, unfortunately, was not giving me long-term remission. So what do you do when you basically run out of options? And so you have to think about a novel approach. And so I went back into my clinical data, and I wanted to identify a signal that could help me to think about a drug that could maybe be used. And so I looked at the cytokine data, looked at the levels of 13 cytokines in the weeks and months leading up to my flare. And I found that VEGF, a marker of angiogenesis or blood vessel growth, and soluble IL-2 receptor, marker of T cell activation, were both quite elevated in the weeks leading up to my relapse. And I was able to confirm that using an orthogonal approach, serum proteomics and then flow cytometry, confirm that VEGF was in fact elevated, that there were in fact activated T cells. And then I thought clinically about clinical correlates. So I knew that my lymph nodes had a lot of blood vessels in them. That's part of the diagnosis of Castleman disease. I knew that my skin had these cherry hemangiomas on them. That's actually also part of the diagnosis of Castleman disease. Cherry hemangiomas would be potentially related to blood vessel growth. And I knew I had a tremendous amount of fluid accumulation, which can also be related to VEGF. And so the question became, is there something central that could be involved in cytokine production, T cell activation, and VEGF production that we could target with a drug? And from a series of pathway analyses that I ran on the proteomics data, so looking for what are the kinds of proteins that are elevated in my blood during flare versus remission, it honed me in on a pathway called the PI3 kinase AKT-mTOR signaling pathway. But as I shared during my lunchtime talk with a small number of your classmates, I needed to confirm that in some way. I needed some sort of orthogonal platform to validate that. So I did immunohistochemistry, where basically you have an antibody against a certain protein. And in this case, it's against phospho-S6. Phospho-S6, or the S6 protein, is phosphorylated by mTOR. And so if the mTOR pathway is active, there'll be increased phospho-S6. So this is a nice readout for mTOR activation. If mTOR, a key signaling pathway, was in fact activated, like my proteomic data suggested, then the mTOR pathway should be turned on and there should be more phospho-S6. So I ran this experiment on a bunch of normal tissue, and so brown is positive, blue is negative. And so in this normal lymph node, you can see that most of the cells are staining blue, so they don't have mTOR activation, they don't have phospho-S6, but there are certainly some cells that are staining positive and certainly do have some mTOR activation. And this is my lymph node, and you can immediately appreciate a huge difference in the amount of mTOR activation. There's just, you don't even need to run statistics when it's this strong. I mean, it just is almost in black and white that the mTOR pathway was really highly activated in my samples. And the important thing about mTOR is that mTOR is actually really important for VEGF production. It's important for T cell activation. It's important for the production of a number of cytokines. And so we thought we had found something that could potentially be involved in all of these. Maybe mTOR being on could explain why all these things were happening. And one of the best parts about mTOR is that there's a drug called sirolimus that's been around for 30 years that inhibits mTOR quite potently. And so I started myself on sirolimus based on these data and based on the fact that we were kind of out of options. And today marks 81.19 months since my last relapse. I mean, you can see from the bar graph that most of my previous remissions were quite short, particularly compared to this remission. And I say 81.19 months because I know that I can't round up. I don't know if I'm going to make it to 82 months that I've been in remission, but I also refuse to round down because my team and I worked so hard for every fraction of a month for me and also for other Castleman's patients. The New York Times called this "Doctor Cure Thyself," which I think is a bit of an overstatement. It should probably be doctor helping himself a little bit right now and hopefully for a lot longer. But I don't think that would have fit on the headline. Importantly, it did give Caitlin and I time to get married. And so we made it to our wedding day, May 24th, 2014, which is really all I could think about. And you can see in the picture, my hair grew back just in time. That's never before been cut hair that you see in that picture. And it was so special that we could make it to that day. And then just a couple years later, or a couple years from now, so a few years after we got married, we had our daughter, Amelia. So this is a picture of Caitlin, me, and Amelia together right around her first birthday. And she just celebrated her second birthday. And she really brings us so much joy, and really never thought that that I would be able to see a day like this. So you guys all read the paper from JCI highlighting our discovery of mTOR as being important and the use of sirolimus in 3 patients. We've since then gone on to show that mTOR is active in a larger number of patients, so 26 Imcd patients, and then also identified a potential driver that could explain why the mTOR pathway is up, and that's increased type 1 interferon signaling, increased responsiveness to type 1 interferon. And so we're starting to hone in on why mTOR may be up, and hopefully that'll help us to get to better therapeutics, because we have seen, this is a picture of Joey. He was one of the first pediatric patients we treated with sirolimus. He had a really, really positive response, and you can see him here. He was very sick, and then this is him with his siblings doing really well. But we've also seen patients like Lisa and Sergio and a number of others where sirolimus did not work for them. Despite chemotherapy, despite IL-6 blockade, despite sirolimus, we were not able to save their lives. And so we've still got a long ways to go. I'm here today based on our research, but we have a long way to go to make sure that a lot more patients are here based on our research. So where are we today in 2020? Well, on the screen, you can see where we were in 2012, and then I'm going to highlight where we are today. So over the last 8 years, we've raised about $1.4 million for research, which has led external groups to then invest an additional $10.1 million into research studies. So over $11 million has gone into Imcd research. The federal government gave the first NIH grant to our lab a couple of years ago. We now have a registry with over 1,200 patients. We have one unified collaborative network that has a unified research platform and agenda. We've got a biobank. We've made progress, though many questions still remain. Thankfully, there's a diagnostic criteria now. There are treatment guidelines. We published the first predictive biomarkers, the first blood test you can run to predict whether you're likely to get better with one drug versus another. Now there is one FDA-approved drug. That drug, siltuximab, went on to get FDA approval. And there's now one drug in development, the drug that I'm on, sirolimus. And so we've made a lot of progress, but we still have a long way to go. About 5 years ago, after I finished up business school, I joined the faculty at Penn. I'm the associate director of Penn's Orphan Disease Center, and I started a center called the Center for Cytokine Storm Treatment and Laboratory. Many of you all would have heard the term cytokine storm in relation to COVID-19. And so we started a center several years ago for cytokine storm research and treatment. And of course, that meant that we became quite interested in COVID when it first arose in the US. I wanted to highlight here, this image is trying to highlight there are about 10,000 human diseases. And out of the approximately 10,000 human diseases, about 1/4 of human diseases have an approved drug. So there are about 2,500 to 2,700 drugs that are approved for about 2,500 to 3,000 of the of known human diseases. But 3/4 of human diseases do not have a single approved drug. And so my case is an example of teaching an old drug a new use. So this is a picture of an old dog reading a book about new tricks. And I think it's really important, and my lab in particular is focused on looking at drugs already approved for something and thinking about new ways they could be useful. We're big proponents of not reinventing the wheel. And over the last couple of years, we've been partnering with the Chan Zuckerberg Initiative to try to scale what we've done for Castleman disease to other rare diseases. I don't have very much time to go through this, but I do want to highlight this concept of drug repurposing. You need to identify and validate candidates for drugs that might be helpful to be used in a new way. You then want to look at real world evidence. So are patients like me getting the drug off label, meaning that a doctor is just giving the drug for something that it's not approved for? And then you want to start thinking about interventional trials where you can see how well is this drug actually working or not working, and then move forward to adoption in clinical practice and FDA approval. So when COVID really emerged in March, we decided that we wanted to be a part of this whole process of identifying new candidates for therapies, but also tracking off-label and experimental drugs to see what drugs seem promising and should actually move forward to randomized controlled trials. So anyone can visit our website at cdcn.org/corona, and you can find it's the largest database in the world of drugs being given to COVID-19 patients. So amazingly, we've had over 60 volunteers. Many of them are medical students from Penn, from Emory, from Yale, from universities all over America, medical students who volunteer their time to go through published studies to pull out data on what drugs have been given to COVID-19 patients and whether they've worked or not worked. The numbers now, I think we're over 250 drugs that have been given to the first I think we're now over 60,000 patients in our database. And these are three of the top drugs most frequently used. It's open source. Our primary users of this database right now are the FDA, Department of Health and Human Services, Google Health, and also the Parker Institute. And so what you'll notice about those groups is that those are the groups that are funding randomized controlled trials. So groups are using our database to see what looks promising, so then they can decide this is the drug that looks really promising that I'm going to spend $10 million on a clinical trial of, or $100 million on a clinical trial of. So our database is really serving as a learning ground, a proving ground for drugs before they move forward to large clinical trials. We published our first paper back in May and have a few more coming down the pipeline. I really would be delighted if anyone from your class or colleagues, friends of yours, would be interested in joining up our effort. We've got some medical students who probably spend a couple hours a week, and we've got others who spend many more than a couple hours, maybe over 10, 15 hours a week, going through these published studies and extracting out data. So we'd be delighted for anyone from your class or colleagues to join up our effort as volunteers. Don't have too much time to go through this, but just to say that we learned a lot from this initial effort. It's really clear that in the immune response, you can either have too weak of a response or too strong of a response. And what we've seen with a drug called dexamethasone is that if you're late stage in your COVID course, then dexamethasone is very effective at suppressing a hyperactive immune response. But if you give dexamethasone early in the immune response when it's not yet strong enough, you can actually cause worse outcomes. And so you really want that middle ground where you're not too strong, not too weak. Dexamethasone can actually make it worse off if you start it too early. Whereas a drug like interferon beta seems to be effective if you give it early on, but if you give it too late, it causes more harm than good. And so this is an example of the challenges that we're facing with COVID and trying to find the right drug for the right patient at the right time. So this is my last slide. I just want to close with a few lessons that are really important. The first is that hope can have many different forms. So I highlighted at the very beginning of my talk that sign at Duke, there is hope, made me hopeful. It made me say, okay, we're in the right place. This is one kind of hope. But then I learned over the course of my journey that if we're going to hope for something, we should really turn our hope into action. And so I think that the hope that inspires action, I think, is the most important kind of hope. The next is how important laughter can be. For all of you watching, I hope that none of you are ever confused as your father's pregnant wife. But if you are, I hope you can find some humor in that and all of life's challenges. I think that there are some really tough times that if we don't laugh, then I think we'd cry. And I think sometimes laughter can be really powerful. It's also really important that we communicate our limits to our patients. I would have never gone on the journey that I went on if my doctor hadn't been very clear with me that I was out of options, that it was unlikely that I was going to survive given the state of things. And so being clear and communicating is so important. The fourth is that solutions may be hiding in plain sight. The drug I'm on, sirolimus, was at my neighborhood CVS for all the years when I was in and out of the hospital, but no one had thought to try it. How many more drugs are already out there that we just need to match? And the last lesson is that it really takes an army. I mentioned earlier that my book is called Chasing My Cure, but it really should be chasing our cures because it really has been an army of people working together. And I really hope you all will be a part of that army to help to spread the word. My book is probably 1/3 focused on my journey of being a medical student, separate from my journey trying to become a scientist and pushing forward the science as an organizer, but really being a medical student, what that's like. And so I think that it would be helpful, and I think that you guys would probably enjoy checking that. And I hope that it would be inspirational to all of you as you move forward in your journeys. So on my slide here, which I'll leave up, you got my email address, you got links to websites you can check out. But thank you guys all so much for your time today.
Okay, so we have time for some questions, and I noticed that some came in through our Zoom Q&A. So I'm going to let Dr. Fajgenbaum go through some of those, and then if students in the auditorium have any questions, you can either type them in or come on up and ask them via the microphone.
Yeah, there's some great questions already in the Q&A. I'm really look good. So the first one, have there been any developments in the search for biomarkers that could predict IMCD since 2012? So we have made some good progress on biomarkers that can predict whether you're likely to get better with one drug versus another drug. We have one paper we published a couple of years ago, another paper currently under review. So there are a few blood tests you can run, and it'll give you a good sense for whether you're likely to get better with IL-6 blockade or not. Unfortunately, we don't have many good alternatives for people who are unlikely to get better with IL-6 blockade. And so sirolimus, the drug I'm on, is one alternative, but we know that it doesn't work uniformly. So though we can predict who's likely to get better, we really need better options for the people who are predicted to not benefit from these drugs. So then there's another good question. What do we think are the pros and cons of using my own data in my research? Your story is important and inspiring, related to Castleman's awareness and the value of life. Thank you so much, Mary. I think that the pros of doing your own research is that you probably you have a vested interest, and so you want to get the right answer. I think the cons are the same thing, is that you have a vested interest and you want to get the right answer. And so that can potentially lead to bias. And so it's not a usual situation to do research on your own samples and your own data. But if for any reason any one of you are in that situation, I think it's really important to assemble a really good team of people so that it's really not just you on your own, and that you really do have people who can look at the data in a different way and can ask questions. Oh, and then there's another question to clarify. So this is predicting the onset of Castleman disease. So we do not yet have any biomarkers that you can run on someone who's healthy and to say you're likely to develop Castleman disease in your future. I hope we'll get there. I suspect there likely will be a genomic signature where you can say, based on your particular genotype, we think it's likely that you could develop Castleman disease. We're unfortunately just not there yet. Question about the mTOR pathway. Are there additional methods to read out mTOR activity beyond phospho-6 expression? Have you assayed for these markers? Yes, there are. We published a paper in Blood about 2 or 3 months ago where we used 3 different readouts for mTOR activation. So one is phospho-6, another is phospho-4 EBP1, and the last is phospho-S6 kinase. And so those 3 readouts are really nice to use because as we talked about earlier, random things can happen randomly. But if you look at the same thing from 3 different angles, you can start to feel a little bit more confident that you're seeing something real. And so we did that for that paper. And thankfully, it showed consistency across the 3 assays. So then there's another good question about how does IMCD affect the body's response to other diseases when someone's in remission? So there isn't really enough good data on this as to how someone like me would respond to, say, COVID, or how someone like me would respond to any number of other conditions, because there just hadn't been very much research before we came on the block about 8 years ago. But I can say that those of us with IMCD are very concerned about what it would be like to experience COVID and whether that would trigger a cytokine storm. So I run a center that has about 20 people involved, and I'm the only one who's never, since March 13th, I have not been back in the lab and back in our center in person. I'm virtually leading things, and I'm really under lockdown. So we're doing everything we can to kind of prevent that natural experiment. But we don't know yet how someone like me would respond to COVID. Let's see, is there any genetic factor involved? Unfortunately, we're not there yet. Hopefully we'll get there soon. But we are doing a large amount of genetic sequencing right now. We're both doing sequencing of lymph node tissue to look for what are called somatic mutations. So mutations that occur over the course of life, like you see in cancer. And if we find those, now it suggests this is more like a lymphoma. But we're also looking at germline mutations, so variants in your germline, what you inherited from your parents, to see if there are actually germline inherited variants that predispose someone to Castleman's. Let's see, you mentioned patients who didn't respond to the same drug you're on or any other treatments. What other drugs might be coming into play or targets for treatments? Great question. So more recently, there have been 2 other pathways that have emerged. One of them is a pathway that we discovered as potentially being important in Castleman's, and that's called the JAK-STAT signaling pathway. So there's both JAK1 and 2 are really important for many cytokines to signal. So cytokines can't cause an effect unless they bind to a receptor on a surface, so the cytokine receptor, and then they work through a number of signaling pathways. So importantly, JAK1 and 2 are key for like dozens of cytokines. And there are drugs that will block both JAK1 and 2. And based on our lab research, we think it's promising to think about using a JAK inhibitor in these really refractory patients. And actually, I just reviewed an article for a journal that looks like someone's treated the first Castleman's patient with a JAK inhibitor. The other pathway is the MAP kinase pathway. Based on some genomic data that we have, we think MAP kinase could be another therapeutic target, but unfortunately, there are far fewer drugs that block MAP kinase than there are JAK. I was interested to see a few Castleman's patients have had whole exome sequencing. Have you considered a larger scale exome sequencing project? So we're in the process of getting a cohort of 300 patients that will have whole exomes done. We'll do whole exomes, plus we'll be doing these SNP arrays to look for variants in the non-coding region of the genome. And we're really hopeful that that'll be helpful for us. Gosh, these are great questions. Under the impression that pharma companies don't get to charge more when it's discovered that one of their drugs is useful for another indication. That's right. If this is the case, how can we play a role in incentivizing pharma companies to devote resources like money and power to look for outcomes of off-label drug use? You just nailed the biggest problem that we have on its head, and that's that currently there really are no incentives. So if a drug company has a drug that's approved for one condition, the reason they got it approved for that one condition is because they felt that was the most likely market to benefit from the drug. And it's the biggest and most likely, that's why they picked that disease. But as you say, there are other diseases that it might benefit from. Those other areas, they may be very rare. So let's say it's Castleman's has 1% as many people have Castleman disease or have some really common type of malignancy like multiple myeloma. So a company makes a multiple myeloma drug and it might work in Castleman's, but they have very little incentive, because if you treat every Castleman's patient with this drug, that's only going to be about 1% of the whole myeloma population. So they're better off just focusing on myeloma. So that means the incentives aren't there for pharmaceutical companies, but that's where we really come in as physicians and scientists, where we can do the work ourselves. So the work we've done with sirolimus has been really independent from the pharmaceutical company, or not just really, but completely independent from the pharmaceutical company that owns sirolimus. So we've done all the work. We've gotten federal funding to pay for clinical trials. We've received grant funding to do the scientific work at the bench. So we have an opportunity. Unfortunately, we as scientists and physicians are actually also a bit de-incentivized to look at old drugs, because we're incentivized to come up with some new cool drug or new cellular therapy that has intellectual property. That's what we're incentivized to do, because our institutions love it when we do that. But I think that we should fight the urge to develop new and exciting new things. I think people need to do that. But the reality is that out of every 100 exciting, cool new therapies, only a few of those are actually ever going to get approval for anything. And so I think that we should make sure that we as physicians and scientists really dedicate time towards existing drugs. Let's see. There's a couple more questions typed in here. I think we can get to both of them. So personalized medicine is certainly one of the causes you've championed. The most recent article that you shared highlights the heterogeneity in cell types and gene expression in Imcd. My question is, to what extent has this observation and the observation that sirolimus does not work in all cases encouraged you to go back and interrogate other pathways beyond mTOR? It has really encouraged us to look at other pathways beyond mTOR. Just as you say, it's very clear that mTOR is not the problem for everyone, because we know you can block mTOR with however much sirolimus you want, and these patients have not benefited. We know the same thing with IL-6. You can block all the IL-6 you want, and some patients will not benefit. So we're very much open-minded about other pathways. And just like in COVID, where there are a lot of promising leads, it's going to kind of come down to the clinical trials to where we actually can say, yes, it works or it doesn't. Let's see. Hi, Dr. Fajgenbaum. Thank you for sharing your story. I was wondering if your experience as a patient shaped the way you look at the practice of narrative medicine. Yes, absolutely. As a patient, I learned that such a small fraction of my journey is actually captured in my medical chart. So my doctors, summarize what I've gone through. They give the high points, but there's so much to my journey that never gets captured in my medical record by physicians. And so I think that's where narrative medicine really can come in, to share the full story. One really good example that I didn't highlight is around cherry hemangiomas. I mentioned that I had these blood moles all over my body that were popping up when I was sick, and they would shrink away when I got healthy. But no doctors noticed them. No doctors asked about them. I kept being like, what about the blood moles? And they kept saying, your liver and your kidneys are shutting down. Forget about the blood moles. But what I found by the fourth relapse was that actually these blood moles were a really good indicator of an impending relapse. So when they started growing leading up to my fifth flare, I knew I was about to relapse because I could visibly see my blood moles growing. That's something that none of my doctors had asked about, none of them had observed. And we held a patient's symposium recently and asked about this. And about 2/3 of the audience raised their hand and say, oh yeah, I do have these blood moles. And oh, I guess now that you ask about it, I guess they do kind of grow before I have a relapse. And so this is an example where narrative medicine and sharing your story beyond what's asked in a medical setting can be really powerful. Question, am I absolutely convinced that sirolimus is the answer for Castleman disease? Was I ever worried about putting most, if not all, our eggs in the mTOR basket? So I am not worried about putting all of our eggs in the mTOR basket, because we have not put all of our eggs in the mTOR basket. Thankfully, we've been doing a lot of parallel projects. There are about 18 studies that are currently underway, and I think 2 of them are focused on mTOR. And so we've thankfully been quite diversified about looking at other pathways. And frankly, I'm glad we did, because we do know that this drug doesn't work for everyone. Let's see. So there's a question here about remaining resilient even during difficult and challenging times. I mean, I think I started out my talk by sharing my kind of rock bottom point. And that's where I didn't think I was going to survive. I didn't think that I could keep fighting, and I wanted to give up. And I think all of us go through those low points at times. And I mean, here I am, my daughter's just in the other room. I'm doing well on this drug. I've got an amazing wife. If I'd given up back then, I would never have had any of this. The patients that have benefited from diagnostic criteria and treatment guidelines and sirolimus and siltuximab, these patients would not be benefiting if I had decided to give up. And so I think that it's really important, during really tough times, to remember, to keep fighting, and that, really amazing things can happen in science when you turn your hope into action. Now, if you just wait and hope that someone else is going to figure something out, it's probably not going to happen. But if you do turn your hope into action, then you can make a lot of progress. So there's a question here. It's not a question, it's a comment. I just recommended your book to my fellow undergrads who plan to go to med school. I think that's awesome. As I said earlier, the book is, it's a lot about my journey chasing my cure, but it's also about my journey as a medical student and what that's been like. And I think that you guys all would enjoy it. Oh, wow. More questions are coming in. Let's see. There's a comment. Really appreciate the commitment to drug repurposing. Wondering how you perhaps speak further about navigating engagement with industry members who may be looking to develop novel mechanisms, maybe doing so at a higher financial price to the medical system. So I think it's important that drug companies work on developing novel approaches. I think it's important that researchers work on developing novel approaches. My perspective is just that while they're working on novel new ways to target new things, I'm going to focus, and our center is going to focus on looking at existing drugs and using them in new ways. Because like I said, we can't halt innovation. We want these new drugs to be developed because frankly, 15 years from now, these new drugs are actually going to become old drugs. And so we need new drugs to be developed. But in our case, we just want to focus our attention on drugs that already exist. It's not that I want to disincentivize other people from doing the work. We need to figure out ways to incentivize drug repurposing. Anything doing with my health that was helpful during my journey, anything that shaped my perceptions of the practice of medicine. I think that one thing that I would say is that as a medical student and previously as the loved one of a patient, I think that I felt like doctors were the only people in the healthcare system. And then I quickly learned as a patient, the importance of all of the supportive team around doctors. So nursing assistants, aides, certainly nurses, PAs, NPs. It's really a team effort. And I think that it took being a patient to fully appreciate that, how much a team effort is. And then it really took being a patient searching for a cure to realize just how important research is. I mean, I think as a doctor, you just kind of know that these drugs exist and you know that they're in your arsenal, but you don't always appreciate just how much work was done on the research side just to make that drug come into existence in the first place. You take that for granted. And so of course, I definitely do not take that for granted. Do I believe that personalized medicine has an application in IMCD, i.e. readouts of biomarkers and selecting appropriate targets? Yeah, I mean, we're already there. You can run 4 blood tests and it'll give you a good sense for whether you're likely to benefit from siltuximab or not. And so you could say, take it, don't take it. Unfortunately, we don't have as many backups. So when people don't benefit from siltuximab, what we need now is, okay, you're likely to benefit from siltuximab, take this. Okay, based on your biomarkers, you're likely to benefit from this other than siltuximab. Right now, it's a bit binary. So hopefully we'll get there. And I do believe that it's in front of us. So it looks like this is the last question. Thanks so much. Is there anything you can think of that has been different in my life, lifestyle, environment, diet, during this remission that could have been that's different than my previous remissions other than medications? It's hard to really pinpoint any one of those things. I've been married during most of this remission. I don't know if that's had any sort of a positive effect on health. Certainly, it's been a positive effect on the mental health, but I don't know if I would attribute it to this remission. It's hard, and I think that this is an important question that you think about when you take care of patients, and that's that you have to take care of them holistically. And so it's not just what drug are they on, but you want to look at them from the whole perspective. And I've thought about it a lot, have not been able to pinpoint anything other than the sirolimus. So I think we got through all the questions. I know we're about 10 minutes over. Is there anything else to hit on before we wrap up, Leslie?
No, it looks like students in the room have been entering their questions as well. So thanks
Nice.
so much for going through all those questions for us and spending the extra time. We all really appreciate it. Excellent talk.
Oh, thanks.